Last module, we learned about how neuron communication within a neuron is an electrical signal that is transmitted by changes in the electrochemical gradient propelled by sodium and potassium moving across the membrane of a cell. For this module, we are going to zoom in at the connection between the end of the axons of the neuron sending the electrical signal and the dendrites of the neuron receiving the signal. What happens when two neurons meet? How do they communicate? Do they use an electrical chemical signal? Well, it turns out that the way two neurons communicate is a little bit different than the way a single neuron communicates within its own structure. So in the sending neuron, remember, it's receiving its own signal from other neurons further up the stream. The dendrites collect that information, it gathers at the axon hillock, and then we get a signal traveling down the axon to the terminal buds. Right at the axon terminal, we have a gap between the dendrites of the receiving neuron and the terminal axon of the sending neuron. This gap is referred to as the synaptic cleft or the synaptic gap or the synapse itself. This space is such that an electrical signal can't actually travel between the two neurons. Instead, what the two neurons use to communicate is a chemical signal. A protein is released from the sending neuron into that gap, into that space. It moves across that space and connects to receivers or receptors on the receiving neuron's dendrites, which then causes small changes in that neuron that may or may not lead it to fire down its own axon shortly afterwards. Many neurons create specific proteins that are designed to transmit this signal between neurons in the synapse. Because as scientists, we like to come up with creative names, we name these proteins, these chemicals that transmit signals between neurons, neurotransmitters. Neurotransmitters are proteins created within individual neurons that, when released, either increase or decrease the likelihood of other neurons further down the chain firing. These neurotransmitters are stored in the axon terminals at the end of each neuron and are contained in what are called vesicles. When an action potential reaches the terminal bud, these vesicles are activated, causing the release of these neurotransmitters. Let's take a closer look at the process of how these vesicles store and release neurotransmitters. There are 5 main steps to the vesicle cycle that I want us to focus on. The first one is loading, the next one is docking, the third one is priming, the fourth one is fusion, and the last one is translocation and endocytosis. So let's take a look at loading first. The first thing to keep in mind is that vesicles are simply cell membrane. They are spheres of cell membrane that have been brought into the neuron through the process of endocytosis that we'll talk about at the end. So these vesicles are spheres of cell membrane that are designed to pull in specific kinds of neurotransmitters through a process called active transport. These are proteins that span the membrane and use ATP and our energy to act as little vacuums, sucking up neurotransmitter from the environment. And they are going to suck up specific types of neurotransmitter. So each vesicle is going to store a given type of neurotransmitter. It is an active process. It sucks it up and stores it for later release. Once a vesicle has all of the neurotransmitter loaded, it moves down to the membrane of the axon terminal. What this vesicle is going to do is it's going to dock to the inner membrane of the neuron and prepare to release its stored contents. Docking occurs prior to any electrical signal being received. It is a process where the proteins on the inside of the membrane of the neuron bind to proteins on the outside of the vesicle. Docking is the first step towards releasing the neurotransmitter, and vesicles must be docked before a neuron can release that neurotransmitter into the gap. Priming is the final stage prior to release. This is when the vesicle begins to integrate with the membrane of the neuron. Remember, the vesicle is membrane. right? So it's very easy for it to integrate with the existing membrane. So what it does by priming is the proteins on the outer part of the vesicle get pulled back by the proteins on the inner part of the axon terminal and begin to pull the vesicle into the membrane. At this point, the vesicle is ready to release the contents of its neurotransmitter as soon as an action potential is received. When the action potential travels down the axon and reaches the terminal bud, this causes an influx of calcium into the terminal bud and is now going to initiate fusion. Fusion occurs when the vesicle fully fuses with the membrane of the neurotransmitter. Remember, the vesicle is just membrane. And so when it fuses, it now becomes a part of the membrane once more, and all of the contents gets dumped into that synaptic cleft, ready to transmit its signal to the neuron on the receiving end. So now we no longer have a vesicle, right? The contents have been dumped, the vesicle is fused with the membrane, and we no longer have a vesicle to fill up again and start the cycle. Well, how do we create that vesicle? Again, as we said, it is created from the membrane of the neuron through a process called endocytosis. In endocytosis, proteins scaffold to the inner edge, the inner side of the membrane, and they grab on to the membrane and they actually suck it up into the terminal bud. So these proteins are actually grabbing a big chunk of membrane from the terminal edge of the neuron and forming it into a sphere and moving it back up into the terminal bud, ready to be loaded with neurotransmitter once more. And that's where the vesicle comes from, is that endocytosis. And that's why the vesicle is simply membrane, because it literally is a big chunk of membrane that is just pulled off and then formed into a sphere and is ready for the process to start over again.